Welcome to Plants, People, Science, a podcast by the American Society for Horticultural Science, where we talk about all things horticulture. In today's episode, we discuss cider and the amazing science and people that bring us this wonderful drink. Today, we have a very special guest, Dr. Greg Peck, who is an associate professor at Cornell's School of Integrative Plant Science. In 2018, the American Cider Association named him Grower Advocate of the Year, and he teaches several cider and orchard management classes alongside his fascinating research. Dr. Peck, welcome to the show.
Hi, Sam. Good morning.
Hey, Dr. Peck, this is Kurt Rome. I got a question for you. For the conversation today, would you like us to call you Dr. Peck, or would you like us to call you Greg? What are you most comfortable with?
Let's use Greg. I think a nice casual tone. And okay if I call you Kurt and Sam?
That'd be great.
Absolutely. So, Greg, in your words, what do you do?
Well, yeah, that's a great question for an academic. We'll be here for days. So my official job title with Cornell is associate professor. I have a 60% research appointment, 30 teaching, and 10 extension. And within my research program, I do several projects. We're going to talk today about the cider research, but I also work in organic apple systems, some work in sustainability, and also crop load management for apples using some modeling work with pollen tube growth.
Greg, today I think we'd like to focus in on the part of your work on cider. So, cider is kind of a short word. It's a small word, but it seems to have kind of big implication, big meaning. When we say cider, what do we mean? What do you mean when you say cider?
Great question, Kurt. Because it does have a lot of different meanings. And so I think traditionally in the United States, cider meant a sweet, unfermented juice that came right off a press. It was usually brown and cloudy. And in the northeastern U.S., it was a big part of a lot of the agritourism. Traditionally here, the U-pick operations, and you'd go and pick apples, get cider apple donuts, unfermented juice. Today, we think about cider more as a fermented beverage. And so cider has a long, long history dating back centuries in Europe. And so it's where apple juice is fermented, usually by yeast, or always by yeast, I guess. And the sugar gets converted to alcohol. And so you have an alcoholic product. And so some people will refer to it as hard cider. Many parts of Europe, they just call it cider. And there's some back and forth in the industry whether hard cider has this kind of negative connotation, hard being something that maybe isn't a positive attribute. And so there's some producers say, we just want to call it cider, not hard cider. Today, in our conversation, when we say cider, we are talking about the fermented product.
Okay, thanks a lot. In preparation for today, I was reading several of your articles, and you've been very prolific in publishing about cider apple production. The cultivars that you talk about are not familiar with me. I don't see them at the grocery store. So what differentiates a cider apple and a good cider apple from, the apples I see at the grocery store, the fresh market, the consumable, the edible and cookable apples?
Right. So cider apples are a very difficult sort of subset of apples to define because all apples contain sugar. And so yeast can convert the juice from any apple into alcohol, and you'd have hard cider. However, there is a subset of apples that have very specific chemical attributes that make them uniquely purpose-driven for making hard cider. And so this might be their sugar content. They have higher sugar, so you get a higher alcohol content. It might be their acidity. And so you get a product that's sharper or more sour, which lends a little bit more of a backbone to the finished cider. Or what's really interesting to us in our program, and what's really interesting to a lot of the cider producers, is they might have a very high concentration of a subset of phenolics or polyphenols called tannins. And these are the ones that the compounds that lend to really make a robust product. It makes it bitter or astringent, depending on the specific types of tannins in the product. But for a fresh eating apple, high concentration of tannin would be very unpleasant. And so oftentimes the cider producers will call these spitters, because once you take a bite out of the fresh apple, you want to spit it out of your mouth as quickly as possible.
That sounds traumatic. I don't ever want to eat an apple like that, but cider is delicious. So there are many factors then that go into making these apples good for cider production. But on top of that, there's also the post-harvest treatments. There's juicing and pressing and fermentation. How does all that change the flavor of cider?
Well, it's very dramatic, and we'll probably over the course of our conversation today try to draw on analogies to wine production, because I think a lot of cider production is very much similar to wine production, right? So grapes have juice in them. You have to somehow extract that juice, and then the yeast will ferment the sugar in that juice into wine, right? And we know though that when you eat a grape, it doesn't taste like wine, right? The flavor composition changes dramatically over the fermentation process, and the finished product, for the most part, especially for the Vitis vinifera, the European wine grapes, are very different between juice and wine. The same is true for cider. And so if you had sweet cider, the unfermented product, right, it's not going to taste the same as the fermented cider, right? And that's because the flavor volatiles change. You might have some changes in aromatics, and you might have some changes in the acid and the phenolic composition over the course of fermentation and also aging. So, when we look for characteristics for what do we want for a cider apple, right? Because I'm a horticulturalist, of course, and that's what we're here to talk about. And so I'm on the apple production side. We always have to kind of think about, okay, well, what is this juice going to be like in fermentation, and what's going to be like after fermentation? And so it's really important, and it's maybe something that when we think about apple quality attributes and a lot of the research that goes into how do we improve apple quality for commercial production, we're also thinking about peel color, fruit size, maybe, and for the breeding angle, might be crispiness, right? We have a ton of apples now that are crisp this, crisp that, or the sugar acid balance in there. But for cider production, we have other characteristics as well that we're really trying to hone in on. So it's different. There's a lot to draw on, but different.
With decades, centuries of experience, of literature talking about how people are making this cider and what sorts of apples work the best, there's a very rich history there. Could you give us an abbreviated history of cider in America?
Yeah. So cider in America, when the European colonists first arrived here, they definitely wanted alcohol, right? Well, other than the Puritans, after the Puritans, maybe. And so, where humans go, typically alcohol follows. And they did try to bring over Vitis vinifera. A lot of those grapevines failed because we have phylloxera, right? Phylloxera is actually endemic to North America, and it's one of our pests that we've released into the world, and it's an invasive pest in many other parts of the world. So a lot of those grapevines perished. They did eventually figure out how to grow apples here in North America, and cider became just part of the landscape in terms of both physical landscape, but also the culinary landscape in the eastern U.S., right? So a lot of farms would have cider apple trees. They would produce cider. It was a very localized production. Sometimes it was used in commerce, but mostly just for household use, or sometimes, just very much so a local thing. Fast forward, centuries, we get up to the late 1800s. We start having a temperance movement. People started to see the ills of alcohol consumption. And we had prohibition in the early 1900s, right? And so one of the famous kind of mythologies about prohibition is that the prohibitionists, the temperance movement, went around chopping down all the apple trees because they were being used for making cider. And it probably happened to some extent, maybe not as much as the mythology says. certainly was some famous pictures of it that really dramatized that. But coming out of Prohibition, what happened is that not only did we have a shift in our kind of view of alcohol consumption, but we also had a shift in our demographics in the country. We had a lot of immigration from Northern Europe, places like Ireland and Germany, beer drinking countries. And after Prohibition, instead of having an agricultural society, an agrarian society, we started to move towards a more urban society. So it's a lot easier to make beer for urban masses because it's a grain, right? Beer is made from a grain. You can store it year round. You can transport that grain. It doesn't have very much high water content. Transport it into urban centers and make beer where the people are. So cider largely died away. If it wasn't Prohibition, it was really certainly those demographic changes that happened, and also the change in our economic structure in our country after Prohibition. So that was in the 1930s, and it really wasn't until the 1990s, 60, 70 years later, that we started to see a few producers say, hey, we can also make alcohol from apples. We should try this. So we had a few cider producers starting in the 90s. And then in the 2000s, we started to see, I think it probably, ties into some of the local food movement, some of the change in demographics where people want to try different things and new foods. And we started to see a resurgence about 20 years ago in cider production. And so now cider is produced by nearly 1,400 different companies around the U.S. in all 50 states, including Alaska, Hawaii, Texas. I mean, there's cideries in all these places. And for retail sales, it's probably somewhere around a $1.5 billion industry.
That's an interesting story. Yeah, even in the state of Arkansas, in the turn of the 20th century, going 19th to 20th century, our large industry was built on either cider apples, brandy apples, or drying apples, which is interesting. So there's this resurgence in the 1990s. Greg Peck is finishing a PhD. How did you get involved in, what caused you, what created the interest in you starting to work on cider apples? Because, I've been in this and there's not a lot of us pomologists that have focused on that market. What got you into it?
It really started when I was at my first tenure track job at Virginia Tech. And I got contacted. I was had a 50-50 research extension split. So I worked a lot with the growers in the state on a lot of different issues around pomology and apple production, mostly for the fresh market. But I got contacted by a few of them, within a year or two of being there and saying, hey, we're getting calls from some of our local cideries and they want us to plant these weird varieties that we've never heard of. Should we do it? And I said, well, geez, I don't know. Let's try to figure this out. And so we started off collaborating with an ag economist. We did some cost of production studies. That was really where we started. Like, what's the economic feasibility? That's what the growers want to know, right? Is this economically feasible? And those results were really favorable. And the calls kept coming to the apple growers saying, hey, we want this variety. We want that variety. All of a sudden it was, we want, so many different varieties because the cider producers didn't really understand the horticultural traits of these. And so they just said, oh, we've read about this, we've read about that, we want these varieties. So the next series of studies, and really it's still ongoing, was about cultivar evaluation and trying to not, we said, okay, well, there's a price premium for these and the demand is high, the supply is low. And now we have to figure out which ones of these cider apple cultivars should we really be growing in Virginia, and continue that work in my current job at Cornell.
Well, that's an interesting start. So now you're knee deep into cider, or barrel deep, I guess. As you have immersed yourself in the science, what are some of the big questions? What are the research questions? What are things I know there can be both grower questions that are researchable, but also I'm sure now that you've been working on this for a decade plus, you see some deeper questions. So what are the questions? Where's the research edge?
Yeah, we have several lines of research in my lab. And many of them are applied just because some of this, no one's really, as you said, Kurt, no one's really touched this topic for 100 years plus in our country or really around the world. We're starting to see a lot more research everywhere, including other researchers in the US, which is exciting. But in terms of my lab, we have questions around, I mentioned the tannins and the polyphenols, a subset of polyphenols that lend mouthfeel and robustness to a product. And so now we're really interested to understand what pre-harvest factors can affect fruit quality, therefore juice and cider quality, right? So I look at a lot of this very similar to, again, going back to the analogy with grapes and wine as what viticulture is to wine production, right? What are the factors that we can manipulate in an orchard that will affect cider quality? So from the very basic side, we just have to understand, like, when do phenolics form in apples, right? There hasn't been a lot of research on that. What controls polyphenol production in apples, right? So there's been research on this, looking at it from a nutraceutical, from a human health perspective. But, we're talking about apples for cider production that have 10 plus times the level of these compounds. And so a lot of the work that we're doing is showing that the phenolics develop very early in the season, typically right at maybe at the point of fertilization, right as soon as cell division really starts, all the way up through that cell division phase, that first 30, 45 days after bloom is when we're seeing a lot of these phenolics develop. And then it kind of is a dilution question, right? As the cells enlarge, they get more water, and they might change a bit in composition. And we're actually studying that right now to try to figure out, they polymerizing more? Are we getting some different types of phenolics developing later in the season? And then on top of that, we ask questions about, what factors might change that concentration or composition. So we look at things like crop load, right? Of course, crop load for apples, crop load is so important. And, we find very strong correlations between how many apples are on an apple tree and the concentration of phenolics in the apples themselves, right? There's a real dilution effect. The more apples you have, the lower the phenolic content. So what causes that? Right? That's the question. And so we're still trying to look at that. We know that it's related to crop load. Is it a source sink relationship? Is it about the carbon reallocation in the tree? Is it other factors, genes being up or down regulated that in response to carbon availability or response to some other factors? We're getting pretty deep into those questions right now in my lab.
Sounds like typical biology. Both nature and nurture are important here.
Yeah, for sure. And so besides for crop load, we also look at things like sunlight. And so we've done a lot of projects over the last 5-6 years, shading different whole trees, shading different organs on the tree. If we just shade an apple versus the apple and the spur leaves, the apple, the spur leaves, and the whole growing shoot. Where, trying to really understand and get at some of these source sink relationships. So that's one line of questioning. We have a whole other line of questioning too, looking at nitrogen, right? So horticulturalists, we're always, go for the big things that are going to control a lot of things in plants, right? Carbon and nitrogen. And so nitrogen, interestingly, is another very specific thing related to cider, which is that, and again, back to the analogy with grapes, is that one of the quality attributes for cider before fermentation is the amount of yeast assimilable nitrogen, or YAN. And this is something that wine producers talk a lot about because yeast need that nitrogen for their metabolism. In addition, if nitrogen content, if the YAN content, the yeast assimilable nitrogen or YAN content is low, The yeast, what they'll do is through respiration, will start depositing electrons on sulfur compounds as opposed to nitrogen, because they've reduced all the nitrogen and then they have to get rid of their electrons right through respiration somehow. And so they'll start reducing sulfur. So reduced sulfur or hydrogen sulfide is the same compound that natural gas companies put into your natural gas so that it smells like rotten eggs. Could be skunky. Clearly, this is a fault, right? This is something that we'd find unpleasant in the finished product. So apples, by and large, have a lower concentration of nitrogen than grapes do. And so this is adding all sorts of new research questions about how do we increase the nitrogen content in apples, but also what yeast strains, right? So we work a little bit with enologists. What yeast strains are there? The yeast strains that are more adaptable to low nitrogen environments and that kind of thing. So a whole bunch of line of questioning along those around nitrogen as well.
I have it's a production question for you. It's not really pomological, but maybe it is. So in the fermentation process, do fermenters add yeast, or are these surface indigenous yeasts? And so the pomological question is, are there factors, are there things that we would do from the production standpoint that would change the indigenous yeast population, the surface yeast population, if that's important in the fermentation process? Again, I'm ignorant about it. You got to fill me in. Tell me.
Oh man, this is a great question, because this is really a hot button issue for the cider industry right now. And and again, analogies to the wine industry. The kind of standard process or protocol would be that the cider producer would add sulfites, SO2, to their juice before fermentation, which would decrease significantly the yeast population, the native yeast population, and then they would inoculate with a commercial strain of yeast and then allow the fermentation to proceed. But there's this whole subset of the industry that wants to do natural fermentation, or spontaneous fermentation would be another name for that, which is allowing, as you said, the yeasts that were either in the orchard or oftentimes in the press room or on the pressing equipment, to allow those yeast strains to become the dominant strain in fermentation. And it's a little bit less of a controlled process, but something that a lot of our smaller scale, I'd say, even some of our medium scale producers are doing in order to allow, either it's a philosophical reason for doing it, right? Because they believe in the more natural process of that, or believe that it is a more natural process. Whereas I think for a lot of our larger scale producers, they want to make sure that they have a reliable product that's going to be the same any day of the year, in any given year, right? And so they want to be able to control it. So we haven't actually done any work looking at what kind of pre-harvest factors might affect yeast diversity in orchards, but there's a few people working on that topic. I think we'll probably discover some interesting things over time. There were a few papers that came out of France a number of years ago where they do some natural fermentations or spontaneous fermentations. And what was really interesting was that the yeast strains in these spontaneous fermentations turned out to be identical to the commercial strains, even though no commercial inoculation happened. And that's because they were the yeast strains that were colonizing everything around. And that's probably why they became commercialized, is because they are outcompeting their cousins and becoming the dominant strain in the environment and also in the fermentation. So it'll be fun to see if the similar story is true for cider.
It's a bit surreal to me, because we're talking and I'm learning about the chemistry and the sulfites and the yeast strains, and you're going so in depth into plant physiology. And yet I've been watching, there's a video online of you giving an orchard tour and you being very involved with growers. And I'm really fascinated by the idea of systems thinking in what you do. I'm curious if you could give your thoughts on the importance of systems thinking in horticulture.
Yeah, thanks, Sam, for that question. I think that really, the idea of thinking about agricultural production as a system is something that, maybe I came into my PhD or my graduate work and my master's and PhD with. Certainly, it's something that evolved during that time, studying organic and conventional systems and comparing them, and knowing that no one practice, no one management um, strategy in an orchard is going to tell you the whole story. And so I've always been interested in the whole picture. And I think that probably comes through in my cider research too, where I kind of touch on different topics. Some of that is thinking like a grower, thinking like what are the growers and answering questions from them, and trying to just keep up with what growers have to think about, right? Because they're always about the system, right? They're never just about one aspect of an orchard. And I think because of the newness of cider apple research in our country, and really around the world, it allows me to kind of apply some of the systems thinking and try to figure out, okay, what are the key questions? Where can we actually make some positive contributions to the science, but also to the producers, and answer some questions for them?
Fantastic. It seems like you must also be teaching this in your classes, right? You teach multiple cider classes, and um, how does that work? Teaching such a varied class to a group of students.
Yeah, so we teach two classes related to cider production specifically at Cornell, and we do it through the viticulture and enology major. And our cider lecture is a 2000 level class. It's open to students in their first through fourth year or beyond. Get a few grad students in there. And it's really a bit of cider appreciation. We do tastings almost every class where they'll taste 3 or 4 different products, or sometimes it's doctored products where we've added certain compounds to display faults, right? We might add a lot of hydrogen sulfide, for example, so they can really get a sense of what that rotten egg smell is. Um, we do some, regional tastings. What are ciders from Spain like? What are ciders from France like? And because they have some unique production characteristics that really typify their styles of cider. It's fun. We do it in a very large lecture hall that was designed for wine tasting and now cider tasting, that allows the TAs to walk between the rows and pour ciders. And we typically get 100, 110 students in that class. What's great about it is I co-teach it with my colleague, Kathy Arnick, and she's a microbiologist by training, and she's an enologist. And so I get to teach everything broadly about the industry, but a lot about apple production up until the point that the apples will get milled and pressed into juice. And then I pass the mic to my colleague, and Kathy then will teach about fermentation and the microbiology and all the things that are in her wheelhouse. So it's a really nice partnership. The students get a lot out of it. I think we, as a general interest, 2000 level class, we get students from all over the university take it. And so we're always, Kathy and I are always trying to slip in as much science as possible. Well, we know that the students are there to taste cider. And then
I want to go back and ask a production question again. I've never worked with cider apples. I do like cider, just to be clear about that. So I'm a consumer, but I've never worked with them scientifically. But as a pomologist, crop load means a lot. And so there's this kind of, and I apologize for the pun, a sweet spot in crop load that we can optimize crop load, maximize fruit size, maximize fruit flavor, which currently means sweetness. So from a cider grower perspective, what is the question? Is it quantity of apples on a tree to increase production, or is it the quality of those apples? And is there a sour spot for cider, just like there's a sweet spot for edible apples?
A lot of the cider apples that producers are trying to grow in the United States come out of Europe. And a lot of these apple cultivars were, and I'll use variety and cultivar synonymously. ACHS likes us to use cultivar in our written work, and the growers always want us to use variety. And so sometimes I slip back and forth just for definition terms. But a lot of these cider cultivars were selected 100 plus years ago for systems where they were being grown in like standard sized trees that might have been 25, 30 plus feet tall, and they were grazing animals, sheep or cows underneath it. Very imprecise sort of production systems. Whatever came in any given year would be what they harvested, and the apples were typically harvested off of the ground, right? So having a propensity for pre-harvest fruit drop was not a negative quality attribute like it is with our culinary apples today. And so a lot of these apple cultivars tend to be very biennial, meaning they'll have a year with a lot of fruit naturally, and then the next year with little to no fruit. And they have these large swings. Well, that's fine in 100 years ago for these very kind of low input systems that were feeding into farmhouse production. But in modern business, we really can't go a year without a crop, or certainly even sometimes if there's a product that has a specific mix of certain varieties or cultivars that they want in there, they have to have those reliably, right? So now the question is, how do we grow these really kind of wonky varieties, right, in a modern production system, in a high density system, and crop them annually? And so it always is, Kurt, a question of quantity and quality, right? It always is. And that's true for culinary apples, it's true for wine grapes, and it's certainly true for cider apples. So some of our recent research that we actually just published in Hort Science, plug for ASHS, we published 2 papers out of David Zakalik's master's work, where we did 3 years of study looking at the different levels of crop load. And we identified crop load, or defined crop load, as the number of apples per trunk cross-sectional area, which is a nice metric to kind of, regardless of the tree size, physiologically, they had the same fruit to vegetative biomass ratio, or, at least that's what we strive for. It's not precise, but it's close. And so we created a whole range of different crop loads on these trees over 3 years, did the same trees, the same crop load. And then we looked at things like the biennial bearing habit, the total yields, and a lot of the fruit and juice quality attributes. So lots of details in there. Encourage your listeners to go read those papers, because I think they're really well written, and David did a really great job on his master's and on those publications from his master's work on those. But the take home was that we could probably crop a lot of these varieties about twice as much as we do our culinary apples. We'll still have some biennial bearing, but we can drastically reduce the biennial bearing. So instead of having these swings with, all production and no production, we get into the in-between. There's still going to be some swings. But when we look at it cumulatively over the long term, we find that the cumulative yield per acre or hectare is larger at somewhere around 9 fruit per trunk cross-sectional area. And that also then will kind of balance between the highest fruit quality or juice quality is at the lowest crop loads. So that's probably impractical from a economic standpoint, right? And then the lowest juice quality, the worst juice quality, is when we overcrop the trees. They had a ton of apples on them, really poor quality. So we, did a little bit of hand waving and kind of looked at all this data from his research and came up with the number. And, we're just trying to give growers some guidance at about 9 fruit per trunk cross-sectional area for a lot of our cider apples. So by comparison, Honeycrisp, a lot of growers are shooting for about 4 fruit per cross-sectional area. Gala, probably about 6 in our climate, fruit per cross-sectional area. So we're going, sometimes up to twice as much fruit as our culinary apples.
Wow. That's incredible. And we will include links to those papers in the show notes. I do want to mention that both of them are in Hort Science. Is that correct?
Correct. Yes.
And they're really good papers. And it's always impressive to me, when you put together a good scientist mentor and a good graduate student, great work happens. So I really enjoyed those papers, and congratulations on your co-author and the student. It shows that a student can really make a difference to our science.
Oh, 100%. I mean, Sam asked earlier about, my job and what I do. And, I said I can talk for hours and days on this. But a big part of what we do is mentoring and training graduate students and training new scientists to get into the field. And it's probably one of the hardest things that we do, but also one of the most rewarding in the end.
Yeah. So wrapping up here, I'm struck that it seems like there's a lot to be excited about. There's exciting research you described, and I'm sure many questions that you didn't even mention that are exciting. There are reasons to be excited with teaching and with the different areas of outreach and research in terms of yeasts and fermentation and so many different fields. I'm curious, leaving off, what are you excited about, Dr. Peck?
Greg. Boy, great question. What's next? What am I really excited by? We have some new work coming out about mechanical harvesting, which is kind of a practice that's been widely used in Europe for harvesting cider apples, and it's something that really helps with the labor issues in our country, right? We have huge political issues and also economic issues around harvest labor. So most of our apples in the US, right, are hand harvested. Might be changing down the road with robots, but we can certainly harvest apples a lot more roughly than we can fresh market apples, right? We can handle some bruising in cider apples because they're going to get milled and pressed into juice. So that's an area of research that we're excited by, is trying to figure out now. We've identified some varieties. We're identifying some management techniques. Now we have to make it cost effective to grow these really unique apples that we know make a really fantastic cider, right? These robust ciders that have these really great quality attributes. But we need to continually look at, okay, how do we do this cost effectively? So mechanization is going to be a big part of that. And I'm also excited by what I'm seeing across the country with cider research. And so outside of my program, a lot of great cider research has happened by Washington State University, and a big shout out to Carol Miles and her program there. We work a lot together on different projects. She's been working a lot on mechanical harvesting over the years as well. But many other land-grant universities around the country are starting to see that their growers are interested in cider apples, that they're diversifying. One really super exciting thing, Sam, is, and I'd love for an anthropologist or a sociologist to get involved in this with us, is we're seeing a return to the farm from the next generation, where for years we always heard, that it's hard to get the farm, the family farm to keep going and have that sustainability to the next generation, because the kids don't want to do it. They want to go and make money doing finance or computer science or whatever. Maybe they want to go into art, but they're not going back to the farm. And we're seeing in New York, at least, there's a lot of examples where the next generation is coming back to the family farm. They say, well, I'm interested in apple production, but I'm really interested in cider production, and are really growing that industry there. So that's also exciting too, is because that speaks well for longevity and kind of generational profitability, right? Like the ability, the cost of startup of a new food or beverage business is so high, and the turnover rate is really high as well, right? Like 50% failure rate. And we haven't seen nearly that high in cider, but there's always that concern. And so that kind of generational and vertical integrated systems or vertically integrated businesses being developed is super exciting. So a little bit outside of my wheelhouse research, but something you can tell I'm excited to see this, because it means like, okay, well, we're doing this work and it's going to mean something down the road too, right? We're not just working on a fad, but we're working on something that is going to be here for the future.
Well, Greg, this has been really interesting, and I really appreciate your time today. Our listeners might be interested in your work, so I think they can find you. You have a very good website. If you Google Greg Peck Cornell or Greg Peck Cider, you'll go to your lab. Now, I want to tell our listeners, and I've waited the whole time, don't Google Gregory Peck because you'll get to the wrong star. We're really talking about the pomological star, Greg Peck, Cornell University, our cider expert in the United States. But it was a great conversation. I learned a whole lot.
Thanks, Kurt. Really appreciate your time. Thanks, Sam.
Thank you, Greg. To read more about cider apples and their history, check out a paper co-authored by Greg titled Growing Apples for Hard Cider Production in the United States, Trends and Research Opportunities. It covers the fascinating trends and challenges in cider production and explains the many differences between American and European cider production practices. This paper is published in Hort Technology, one of the open-source peer-reviewed journals published by the American Society for Horticultural Science. A link to this article and the others we talked about during the episode will be provided in the show notes.
And again, as I said, if you want to learn more about Greg and his work, you can find his bio at the Cornell website, or you can find him featured in an article in modernfarmer.com.
If this interview resonates with you, I highly recommend renewing your membership and joining the American Society for Horticultural Science so you can be a part of interest groups like pomology or post-harvest or fruit breeding. If you'd like more information about the American Society for Horticultural Science in general, you can go to ashs.org.
Thank you, Sam, and thank you for listening today.